Good afternoon once again and welcome to your class in NAPSI 22 Agricultural Biology. So we have discussed last time under Unit 2 on the bioenergetics. So this time we're going to talk about enzyme. So we have mentioned earlier on our subject on bioenergetics the characteristic of enzymes, and we know that they catalyze changes, especially in physiological processes. So once again, what are enzymes? Most enzymes are proteins, so tertiary and quaternary structures. So this looks like the enzymes and act as catalyst to accelerate a reaction. So it will definitely change the substances and not permanently change in the process. So it can always change from one form to another because of the enzyme. So this is the specific figure on how the enzyme will act to catalyze. So we have here um, a substrate wherein there is a specific structure which are fitted to the substrate in the active site code. So this is the enzyme itself, and these are reusable and usually end in ase. So we have the sucrase, lactase, and the maltase. So from the word sucrose, it is if it is an enzyme dot catalyze change or divide sucrose, that's called sucrase, or for lactose, lactase, and for maltose, maltase. So how does these enzymes work? Enzymes work by weakening bonds, which lowers activation energy. So they weaken the band. So this is some a figure on an illustration on how enzyme lowers activation energy for catalyzed reactions. So this is the reactant Y, and it usually forms. And as the enzyme is catalyzed during a reaction pathway, the product is the energy, the total energy decreases as it goes to a reaction. So another figure to show you the energy as it grows to usually it seems like there's a peak and then the decrease of the free energy as it goes to the final product. So we have here two categories. The with enzyme is this colorful purple one, and this the bluish is without enzyme. So the free energy therefore is the activation of the free energy by the reactants is higher without enzyme than the with enzyme as the progress of the reaction occur. So what are enzyme and its reaction? So we have here examples. No? Enzyme without substrate, there is no reaction. An enzyme with substrate like the H2O and the oxygen bubbles products, these are the enzymes with reaction already. And you have here the liver homogenate catalyzed enzyme and the substrate hydrogen peroxide. So the enzyme substrate complex is the one that is introduced earlier, wherein the substrate is fitted and is joined into the particular active site of the enzyme. So the substance is the reactant, and an enzyme acts on it is the substrate. So the active site is the enzyme itself. It's a restricted region of an enzyme molecule which binds to the substrate, as shown on that figure. And we have the so-called induced fit because the active site and the substrate must fit into its structure. A change in the shape of an enzyme's active site is called as the induced fit. It is induced by the substrate. So it may, for the active site may change in form if it is induced, fitted for the substrate. So a change in the configuration of an enzyme's active site with the hydrogen ions and ionic bonds are involved in this process. And this is induced by the substrate, this one. So the substrate itself will induce the fitness of the active enzyme site. And what affects this enzyme activity? There are three factors. We have the environmental conditions, the cofactors and the coenzyme, and the enzyme inhibitors. Let's start with the environmental factors. First is extreme temperature are the most dangerous. We all know that we die at higher than even 50 to 60, 100 degrees Celsius. Then high temperature may denature or unfold the enzyme. So with high temperature, extreme temperature could denature the enzyme. And pH, most likely 6 to 8 pH which is near neutral, will also affect the enzymes and the ionic concentration or salt ions. So we also have cofactors and coenzymes. These are inorganic substances like zinc and iron and vitamins respectively are sometimes needed for the proper enzymatic activity. Example, iron must be present in the quaternary structure for the hemoglobin in order for it to pick up oxygen. And we all know that. That's why persons become anemic if we lack iron, because and as symptoms for that, we usually faint because we lack oxygen. We lack the hemoglobin which picks up the oxygen. So there are two examples of enzyme inhibitors. We have the competitive inhibitors. These are the chemicals that resemble an enzyme's normal substrate and compete with it for the active site. We have the substrate, the competitive inhibitor, and the enzyme. So they just resemble normal substrate. These are not the real or the original substrate, but they have to compete for the active site. So therefore they inhibit the actual substrate that must be fitted for the active site. So the second is the non-competitive inhibitors, inhibitors that do not enter to the active site but bind to another part of the enzyme itself, causing the enzyme to change its shape, which in turn alters the active site. So this time, the competitive inhibitors, non-competitive inhibitors are the inhibitors which doesn't go to the active sites, but the substrate goes to the other side and which alter, and eventually it will block the substrate from the active site. So these are for about the enzymes. And if you have questions, you can raise your questions as you watch the recorded video on this one. So we will further continue on the biochemistry of cells in continuation of our topic. For the biochemistry of cells, we'll just discuss this on the next video. So thank you for watching, and I hope to see you again on the next video.